cell cycle by the one and only Rosales. So before we talk about the cell cycle, let's talk about the cell size limit. So the main factors that limit the size of a cell is the ratio of its surface area to its volume. So the surface area of the cell is the area covered by the plasma membrane or the outside of the cell, and the volume is the space taken up by the inside of the cell, like the organelles and the nucleus, and anything inside the cytoplasm. So once a cell reaches its size limit, it will either need to stop growing or divide. So cell division not only prevents the cell from becoming too large, but it also has a role in reproduction, growth, and in repairing or healing injuries. So cells are produced by a cycle of growing and dividing called the cell cycle, which is made up of 3 stages: interphase, mitosis, and cytokinesis. So we have interphase, which is the first stage and the longest stage of cell division, and it's made up of 3 phases: G1, the S phase, and G2. So the first phase of the interphase is G1. So a cell is growing, carrying out its normal cell processes and its normal activity, and it is getting ready to replicate its DNA in the S phase. So in the S phase, DNA is replicated and it's getting ready for cell division. So in the last phase of interphase, which is the G2 phase, the cell prepares for mitosis and the division of its nucleus technically. And so yes. So here's the cell undergoing interphase. So after interphase, the next stage of the cell division is mitosis. And like interphase, mitosis is made up of phases, but it's made up of 4 phases. So the first phase of mitosis is prophase. So in prophase, the chromatin condenses into these little X thingy-mabobbies called chromosomes. So these chromosomes, or the half of the X of each chromosome, is called a sister chromatid, and they're linked together by a centromere right here. So in this stage, the nucleolus disappears and microtubule structures called spindle fibers form in the cytoplasm. So in only animal cells, another pair of microtubule structures called centrioles form. So this whole structure consisting of the spindle fibers, the centrioles, is called the spindle apparatus or the mitotic spindle. So near the end of prophase, the nuclear envelope, which is this right here, disappears, and the centrioles move to opposite poles of the cell. So here you can see the prophase occurring in the cell. So the second phase of mitosis is metaphase. So in metaphase, the spindle fibers attach to the chromosomes, and so the centrioles are on both sides of the cell. They have to attach to both sides of the chromosome, so they attach to the centromere technically. And once they're attached, the chromosomes line up in the middle or the equator of the cell, like shown here. So here is the metaphase stage in a real cell, and you can see the chromosomes lined up here and they're attached to the spindle fibers. So after metaphase is the third phase of mitosis, and it's anaphase. So in anaphase, the sister chromatids are separated. So to go in further detail, the microtubules of the mitotic spindle begin to shorten. This pulls at the centromere of the sister chromatids, causing them to pull apart. Oh, it's okay. Say bye-bye, okay. And then once they're pulled apart, they move toward the poles of these cells. That's shown here. See the microtubules are shortening and the chromatids are pulled apart. Exactly. Okay, and then the last stage of mitosis, not of cell division, mitosis is telophase. So once the chromatids arrive to the poles of the cells, they decondense or loosen up or relax. They decondense into chromatin. New nuclear envelopes appear, and the nucleoli reappear. Hence the word nucleoli, not nucleolus, means plural, so there will be two. And the spindle apparatus dissolves. See, spindle fibers are withdrawing. So here's the picture, telophase part. You can see. Okay, and finally we have come to the last stage of cell division called cytokinesis. So in cytokinesis, the cytoplasm is divided and it results in two identical cells with identical nuclei, as you can see here. So only in animal cells, cytokinesis is completed by using some type of microfilaments microfilaments to pinch the middle of the two almost separated cells. It keeps pinching until they are separated, and that pinch point, I guess you could call it, is called a cleavage furrow, right here. So the completion of cytokinesis results in two identical daughter cells. These cells are diploid, meaning they have pairs of chromosomes. So here you can see cytokinesis has taken place. And I bet you're wondering, well, how long does the cell take in each of these stages? Well, here's some approximations. But as you can tell, interphase is the longest stage, and in mitosis, four phases, stages, things, the longest of them is prophase, and these other three, metaphase, anaphase, and telophase, are pretty close together. So that's it. That's the end of the cell cycle, and it starts all over with each two daughter cells. It keeps going. Yeah.